Hi everyone and welcome to video 5. So we should now be an expert at what diffusion is and what diffusion means, and we should also be an expert in the cell membrane and what it is made up of. So if you are not and you're struggling with that, you need to go back to videos 4 and 3. So what we've got to do for our starter activity today is to write down the definition of diffusion. If you want the consumables for this video, you need to go to your cell biology, and then you need to go to transport, and then you need to go to activities and consumables, where you can access the osmosis and active transport, which is 2.3. And then you can print off all of the consumables and worksheets to go along with this osmosis and active transport video. So if you pause the video and then you write down the definition of diffusion of how you think it should be written. So the answer is that diffusion is the movement of molecules from a high region of concentration to a low region of concentration down the concentration gradient, and it is a passive process, meaning that it requires no energy. So today our learning intention is to define osmosis, so to be able to write a definition of osmosis, and to explain the relationship between different concentrations of solutions and their effect on cells. So what I am looking for you to be successful is for you to label diagrams on how cells are affected by different salt solutions or solute solutions, answer the questions on osmosis, and stand and deliver what osmosis is and what happens to the cell at different water concentrations. Osmosis is a passive process, which we learned in the last video tutorial. So that means it requires no energy, and that's because it's very, very similar to diffusion, except from osmosis is a special case of diffusion because it involves water molecules. So not everything can move by osmosis, only water. So osmosis kind of sounds like Oasis, which is a liquid, so that's maybe how you could remember that it's water. So osmosis is a special case of diffusion. Osmosis is the movement of water molecules from an area of high water concentration to an area of low water concentration through a selectively permeable membrane. And we've heard of the word selectively permeable membrane before when we were talking about the cell membrane. So the cell membrane is selectively permeable because it will only let selective things through. It will let small soluble molecules through, but will not let large insoluble molecules. So osmosis is passive and it doesn't require energy because it's like a person going down a big steep hill. They don't need to put in any energy. The wheels will just go off the bike down the hill. So it's always moving from high to low down a hill. Okay, so osmosis is the movement of water molecules from an area of high concentration to an area of low water concentration through a selectively permeable membrane. So you want to write the head in osmosis and you want to write this note, and you want to make sure that you're able to say this note, and you could write it out a few times. You could say it to lots of different people via FaceTime, etc. But learn it. So there are various different osmotic effects in animal cells. So if you put an animal cell in a high water concentration, so in pure water, it'll have a different effect on the animal cell to if you put it in a really, really solute solution, so like really, really salty solution where there's lots and lots of salt and a little bit of water. So animal cells change if surrounded by solutions of higher or lower water concentration. So they kind of act a bit like a balloon. So if you were to fill up a balloon with water, what would eventually happen to the balloon? Okay, so what do you think might happen to an animal cell in a pure water concentration, so a high water concentration? So what's going to happen is the water will go from the high water concentration outside of the cell to the low water concentration inside of the cell, and it'll fill the cell until it swells and then it will burst. Okay, just like a balloon, it'll swell and burst. Now, if you put the animal cell, like the red blood cell, in a salty solution or another solute solution, like sugar solution, that just means solute solution just means that it's something that's dissolved in water, so like salt, sugar, etc. So if you put that, an animal cell into that, what's going to happen is the animal cell is going to have the high concentration of water on the inside of the cell. So it's going to move, the water will move from the high water concentration inside the cell to the low water concentration outside of the cell. And this is like if you squeeze the water out of the balloon, it's just going to squeeze and squeeze and squeeze, and then what will happen is the animal cell will shrink, like the balloon will shrink and it will shrivel. So if we have a red blood cell, a normal red blood cell, and it has a 0.85% water concentration, and we put it into a solution that is 0.85% water concentration, then obviously the water concentration outside of the cell is equal to the water concentration inside of the cell. As it was said with the picture before, if the blood cell is surrounded by a solution of the same concentration, there will be no gain or loss of water, so the cell is unaffected. So basically the water will still be moving, but it'll be going in as fast as it is going out, so it will be equal. So the cell will not change shape because there's no gain or loss of water. Animal cells work best if kept in their normal conditions. So this is why they wouldn't put pure water into you in the hospital, and they usually will inject drugs in saline solution. So they'll put salty water into your drip and not pure water, because if they were to put pure water, your animal cell would obviously burst. So they need to put an exact concentration of water and salt in the saline drip so that there is no gain or loss of water, so that your red blood cells will remain unaffected, and same with the rest of your cells in your body. So this is showing you again that if you place a cell, so the red blood cell, in a salt solution with a concentration equal to that of the cell, the water will diffuse into the cell at the same rate as the water diffuses out of the cell. So there will be no net movement of water, and therefore the red blood cell will stay the same shape. So no net gain of or movement of water, so the cell is left unaffected. So if we change the solution, so if we put our animal cell, like a red blood cell, into a lower water concentration this time, so lower water concentration means that there's a lot of solute in it. So it could be sugar, it could be glucose, it could be sucrose. So if surrounded by a solution with a lower water concentration, so salt solution for example, there is a net diffusion of water out of the cell from an area of high water concentration inside the cell an area of low water concentration outside of the cell. So the water moves outside of the cell by osmosis, causing the cell to shrink. So you always want to say where the water is moving from and to. So you'd want to say that it's moving from the high water concentration inside the cell and to the area of low water concentration outside of the cell by osmosis. So this is an illustration of what would happen if you put your red blood cell into a salt solution of greater than the cell's contents. So there's going to be a net movement of water out of the cell, so it's going to move from the high area of water concentration inside the cell to the low area of water concentration outside the cell, which is going to cause the cell to shrink and shrivel. So it originally started as round and now it has shrunk. So if we put our animal cells, like our red blood cells, in a high water concentration, so that means that there's a tiny bit of salt or there's no salt at all. So that means a high water concentration is going to be on the outside of the cell this time. So if surrounded by a solution with a higher water concentration, there is a net diffusion of water into the cell from an area of high water concentration outside of the cell to an area of low water concentration inside the cell. So it's important to see where the water is going. The water moves inside the cell by osmosis and causes the cell to swell and burst. So this shows an animation of what happens if you put an animal cell, like a red blood cell, into a high water concentration. So the cell will be placed in a salt solution with a concentration less than that of the cell, so for example pure water. So this means that our high water concentration is going to be on the outside of our cell, this time, and our low water concentration is going to be on the inside of our cell. And we know that osmosis is the movement of water from a region of high concentration to a region of low water concentration through a selectively permeable membrane. So the water is going to enter the cell. So what will happen is the cell will swell and then it will eventually burst. So the cell gains water, so it swells up and eventually bursts. So osmosis in animal cells is dangerous. So because animal cells do not have a cell wall to protect them, they are in danger if the water concentration outside the cell is too high, because they'll burst, and if they're placed in a salt solution, they will shrink and shrivel. And you can remember this by if you put lots of water in a balloon, it would get really fat. So if you put them in a high water concentration, they're going to get really fat. And then if you put them in a salty solution, beginning with S, they're going to shrink and shrivel. So if you printed out the consumables, you should have this diagram here. So this is a summary of what happens to the animal cells. So because they have no cell wall, if you immerse them in a pure water concentration, so a higher water concentration, the water passes into the cell from a high water concentration on the outside of the cell to the low water concentration on the inside of the cell, causing the cell to swell and burst. If you put the animal cell and you put it into an equal water concentration, then there's no net movement of water, so the cell remains the same shape and it remains unchanged. And if you put an animal cell into a salty solution, so more salty than the cell content, the water passes out of the cell from the high water concentration inside the cell to the low water concentration outside of the cell, and it will cause the cell to shrink. So you're wanting to say where the water is going from and going to, and what's happening to the cell. So again, this is just showing you a diagram of the red blood cells. So if you put it in a pure water solution, where the water concentration is higher on the outside of the cell, then the animal cell will burst. If you put it in the same or equal salt concentration, then the animal cell will stay the same because there's no net movement of water. And if you put it in a salty solution, the water will move from the high water concentration inside of the cell to outside of the cell, and the animal cell will shrink. So next we're going to look at plants and how they differ from animal cells. So before we went into lockdown, some of you will have carried out the potato investigation. So basically we set up different salt solutions and we put cylinders of potato that were the same size, mass and weight. First we had to blot the potato cylinders dry, so to make sure that there was no excess water left on the potato, or that would have made our results invalid. So for example, this is going to show you two types of conditions. So they've got distilled water, which is quite pure water, and they've also got a very strong salt solution in their other one, which are equal volumes. So they're putting their potato cylinders in, and then they're leaving it for 24 hours to see what happens. So after the 24 hours, we should see that potato cylinder A has become bigger, heavier and firmer, and we should see that potato in salt solution has become smaller, lighter and softer. So this is quite similar to what we've been saying. So if we look at the first one, so potato cylinder A, we know that the high water concentration is going to be outside the cell because it's pure water, and we know that the low water concentration is going to be inside the cell. So the water is going to move from the high water concentration to the low water concentration into the cell, which is going to make it become heavier, firmer and bigger. Now for potato cylinder B, which is in a very strong salt solution, we know that the high water concentration is going to be inside the cell this time, and we know that the low water concentration is going to be outside the cell. So the water is going to move from the high water concentration inside the cell to the low water concentration outside the cell through the selectively permeable membrane. So this shows you a diagram of what happens. So if we put the piece of potato into pure water, we've got our water molecules, which is our high water concentration of water molecules on the outside. We've got our low water concentration of water molecules on the inside. So your water will diffuse from the high concentration to the low concentration, and our starch, which is in our potato, will not be able to move. So our water will be diffusion, but our starch can't move. So our starch molecules can't cross the membrane as they are too big. So the potato is going to get bigger, heavier, and firmer as the cells fill with water. And that's what's going to cause the potato to get bigger. Whereas if we put the potato into a highly concentrated salt solution, we've got less water. So we see our salts taking up a lot of the spaces. So the white blobs are taking up a lot of the spaces. So our higher water concentration is now on the inside, and our low water concentration is on the outside. So the water molecules are now more highly concentrated inside the potato, so the water will diffuse from the high concentration to the low water concentration. So the potato gets smaller, lighter, and softer as these cells lose water. So the osmotic effect in plant cells is similar to what happened in the animal cell. So for example, we've just talked about the potato, but plant cells have a cell wall, so they will not burst when they're put in high water concentration, whereas animal cells do burst. When we put our potato in that pure water, it got bigger, but it didn't burst. So if we put a potato cell or any plant cell into the same water concentration, so if the water concentration is the same on the outside as it is on the inside, the water will enter and leave the cell at the same rate, so there will be no net gain or loss of water. So there's no net movement of water, so there will be no change in appearance of the plant cell. So if we have a normal plant cell, we have our freely permeable cell wall, our selectively permeable cell membrane, and our water concentration of our cell sap is equal to a dilute sugar or salt solution. So if we are to place a normal plant cell in a dilute sugar solution or solute solution of any sort, then the water diffuses into the cell at the same rate as it diffuses out of the cell. So there will be no net gain or loss of water. So the cell will remain the same shape and it will remain unchanged. So if the plant cell is put in a high water concentration, so for example distilled water or pure water, the water is going to move from the high water concentration outside of the cell to the low water concentration inside of the cell. And that's going to cause the vacuole in the cell to swell. And when it swells, it's going to push against the cytoplasm and the cell membrane, and it's going to push them against the cell wall. So this cell wall is stopping that plant cell from bursting. And when the cell is in this state, so it's all pushed together and basically quite fat and swollen, it's said to be called turgid. So you need to know that when a plant is put into a high water concentration, it becomes turgid. So high water concentration, turgid. So that word is really, really important that you learn it. So that's when your vacuole swells, pushes against your cell wall, so that'll push your cell cytoplasm and it'll also push your cell membrane against that cell wall, and the cell is said to be turgid. So remember the word turgid. So high water concentration makes the cell go turgid because the water enters the cell. So this is an animation of what we just said. So a normal plant cell is placed in pure water. So you've got your high water concentration on the outside and the low water concentration on the inside. And we know that by osmosis, the water is going to move from the high water concentration to the low water concentration through the selectively permeable membrane. And it's going to then cause this vacuole to swell, and it's going to cause the cytoplasm to push against the cell wall and also the cell membrane. So water diffuses into the cell from an area of high water concentration to an area of low water concentration. The cell gains water and gains turgor. The tissue is now said to be turgid. So we have to remember that when the cell is swollen, it is called turgid. turgid. If we put plant cell in a low water concentration, so for example a solute solution or a really salty solution or a really glucosey solution, etc., what's going to happen is your high water concentration is going to be inside of your cell and your low water concentration is going to be outside of your cell. So your water is going to move from the high water concentration inside of your cell to the low water concentration outside of the cell. So this is going to cause your vacuole to shrink. Now when your vacuole shrinks, it's going to pull in the cytoplasm and it's going to peel the cell membrane away from the wall. And when it is in this state, the cell is said to be plasmolysed. So you need to remember when it gets put into a salt solution, the plant cell is going to become something called plasmolysed. So that word is very, very important. So salt solution, plasmolysed. So this is just an animation of what would happen if you put your plant cell in a sugar solution. So as we said, if you put it into a sugar solution, the high water concentration is going to be inside of your cell and the low water concentration is going to be outside of your cell. So the water is going to move by osmosis from the high water concentration to the low water concentration through the selectively permeable membrane. So that is going to cause your vacuole to shrink and it's going to cause your cytoplasm to peel your cell membrane away from the cell wall. So then you get a plant cell that looks like this, and when it looks like this, the plant cell is said to be plasmolysed. And we need to remember, if you put it in the salty solution or any solution that's a solute solution, your plant cell is going to become plasmolysed. The water diffuses out of the cell from an area of high water concentration to an area of low water concentration, and the cell loses water and becomes plasmolysed, and the tissue is said to be flaccid. So the important word here is to remember plasmolysed. So that's when your cell membrane is peeled away from your cell wall, which you can see there. So you should have this summary note if you printed out the consumables. So you would then want to put and try and fill in the blanks yourself and pause the video. So if you immersed a plant cell in pure water, what should happen is water passes into the cell from the high water concentration on the outside to the low water concentration on the inside of the cell, causing the cell to be turgid. And what turgid means is that the vacuole swells and pushes against the cell wall. If you put a plant cell in an equal concentration, then there will be no net movement of water and the cell will remain unchanged. And if you put your plant cell in a salty solution or a sugary solution, then you'd expect your water to pass out of the cell from the high water concentration on the inside of the cell to the low water concentration on the outside of the cell, causing the cell to become plasmolysed. And that means the vacuole shrinking and pulling away from the cell wall. So this is just a diagram summarising what we have said. So osmosis in plant cells. So if you immerse a plant cell in pure water, the high water concentration is going to be on the outside of the cell and the low water concentration is going to be on the inside of the cell. So the water is going to move from the outside of the cell to the inside of the cell, which is going to cause the vacuole of the plant cell to swell, and it's going to push against the cytoplasm and the cell membrane, which is going to push against the cell wall, which is going to make the plant cell become turgid. Now, if you put a plant cell in a dilute sugar solution, which is equal to the sugar solution in the cell, then there's going to be no net movement of water because the water is going to move in at the same rate as it moves out, so the plant cell will not change in appearance. If you put a plant cell in a concentrated sugar solution, so that there is a higher salt solution outside of the plant cell, then the water is going to move from the high water concentration inside the cell to the low water concentration outside of the cell by osmosis, and that's going to cause the vacuole to shrink, and when the vacuole shrinks, it will pull the cytoplasm and the cell membrane away from the cell wall, and it will become plasmolysed. So we need to remember the word plasmolysed for if it's put into a sugar solution. So now you should be able to answer why do plant cells not burst like animal cells, and you should make a note of this. So the reason being is plant cells have a cell wall, so they're protected by their cell wall. Animal cells don't have a cell wall, so they burst. So sometimes in osmosis questions you will be given the solute concentration, but you'll not be given the water concentration. And it is important to be able to calculate the water concentration from that, because we know that osmosis is the movement of water, it is not the movement of solute. Remember, solute is just anything dissolved in water, so sucrose would be a solute in this instance. So we need to be able to work out the water concentration so that we can see where the water is high and where it's low, so we know where it's moving to. So for distilled water, you're going to have 100% water and you will have 0% sucrose concentration. For 10% sucrose solution, you will have 90% water and you will have 10% sucrose. For 20% sucrose solution, you'll have 80% water and you'll have 20% sucrose. And then for 40% sucrose solution, you would have 60% water and 40% sucrose solution. So as you can see, all of these numbers add up to 100. So this one would have our highest water concentration out of this table, and this one would have our lowest water concentration out of this table. So this is similar to the last table. So this shows us our concentration of sucrose in molar, and then we've got our volume of water in centimeters cubed, which also is in milliliters because centimeters cubed is the same as milliliters. And then we've got our volume of 1 molar dm to the minus 3 stock sucrose solution in centimeters cubed again, which is the same as milliliters. So for 0.0 concentration of sucrose in molar, we have 100 centimeters cubed of water to 0 centimeters cubed of sucrose solution. And then for 0.2, we'd have 80 centimeters cubed of water and we'd have 20 centimeters cubed of sucrose. And then for 0.4, we'd have 60 centimeters cubed of water and we'd have 40 centimeters cubed of sucrose. And then for 0.6 molar, we'd have 40 centimeters cubed of water and we would have 60 centimeters cubed of sucrose. And then for 0.6 molar, we'd have 40 centimeters cubed of water and we'd have 60 centimeters cubed of sucrose. And then for 0.8, we'd have 20 centimeters cubed of water and 80 centimeters cubed of sucrose. And then for the last one, we'd have 1 molar of sucrose, which is our strongest. So we've got 0 centimeters cubed of water and we've got 100 centimeters cubed of sucrose. So obviously the strongest solute concentration would be the one at the bottom, and the lowest would be the one at the top. So this one would have your highest water concentration and this one would have your lowest water concentration, because remember it is your volume of water which is important in determining the direction of osmosis. What you can do now is you can go try and do the salt concentration consumable 3 and osmosis worksheet consumable 4A, and you can pause the video, and once you've completed them worksheets you can send it to your teacher for marking. So you should now be able to define what osmosis is, describe what a concentration gradient is, describe what happens to a blood cell in a high water concentration environment, describe what happens to a plant cell in a high water concentration environment, and why don't the animal and plant cells react the same in a high water concentration. So what you can do is you can pause the video and you can answer the questions, and then I will give you the answers. So I'm going to wait and give you the answers now. So you should have written for define osmosis. Osmosis is the movement of water from a region of high water concentration to a region of low water concentration down a concentration gradient through a selectively permeable membrane, and it is passive, so it doesn't require energy. For the second question, describe what a concentration gradient is. It's anywhere where there is a difference in concentration, so there's a high concentration and a low concentration. For 3, describe what happens to a blood cell in a high water concentration environment. So if you have a blood cell, so let's draw a little diagram because it's easier to put it in a diagram to explain. So you have a high water concentration environment on the outside. Okay, and you have, if it's a high water concentration on the outside, then it must be a low water concentration on the inside. So you then go through the process of osmosis. So osmosis is the movement of water from a high water concentration to a low water concentration through a selectively permeable membrane. So the water is going to enter the red blood cell. So you would want to say that the red blood cell is going to swell and it's going to eventually burst if it is put into a high water concentration environment. Now that is not the same with a plant. So question 4 asks what is going to happen with a plant. So if we have our plant cell and we've got our high water concentration outside and our low water concentration inside, our water is still going to move through osmosis, so it's still going to go from the high water concentration to the low water concentration. And this time your vacuole is going to push the cytoplasm and the cell wall against the cell membrane, against the cell wall. So that's going to cause the cell to become something called turgid. So we would want to say that our plant cell is going to become turgid in a high water concentration environment. And for the last questions, why don't the animal and plant cells react the same in a high water concentration? That is because your plant cell has a cell wall, so the cell wall protects it. Our animal cell doesn't have a cell wall, so our animal cell will swell and burst. Our plant cell with its cell wall will become turgid because it can't burst, because it's got that cell wall to protect it. So well done if you got all 5 of these right. If you didn't get all 5 of these right, you might want to go back to the start of the video and then watch it over again. So an osmometer can be used to measure osmosis. So in this diagram we have a capillary tube and we have our sugar solution, which is in viscous tubing, and we've got our water. Now what should happen is, in this situation, we should have our high water concentration on the outside because we're told that it is a watery solution, and we should have our low water concentration on the inside because we're told that it is a sugary solution. So our high water concentration, well, osmosis should move the water from the high water concentration to the low water concentration. Now if it is pushed into this, there's nowhere else for the water to go except from up this capillary tube. So to measure the rate of osmosis, we would have to time how long the water takes to get to a certain distance. So there are certain ways we could speed up this, so how fast osmosis occurs, and that would be either to increase the concentration gradient or to increase the temperature. So there's 2 questions for you. So explain why the water moves up the capillary tube and how you can speed up the rate of osmosis. We already kind of went over that, so you can pause the video and try to answer these questions yourself. So for the first question, the water moves from the high water concentration outside the viscous tubing to the low water concentration inside the viscous tubing, which pushes the liquid up the capillary tube. And how can you speed up the rate of osmosis? Well, you can increase the concentration gradient by increasing the concentration of sugar, or in any case of any solute, or by increasing the temperature. So in summary, if you write down osmometer, and then you would write an osmometer is a piece of apparatus used to demonstrate osmosis and measure its rate. So as your water moves up the viscous tubing, the level in the capillary tube rises. The rate of osmosis can be measured by timing how long the level takes to move a certain distance. So a lot of you will have been lucky enough to do a potato investigation, but some of you might not have been. So basically it's the experiment where you had your different test tubes with your different solute concentrations, and you put your potato cylinders in each of your different solute concentrations, then you leave them for about 5 minutes or 10 minutes, and then you weigh them again to see what has happened, so whether they've gained weight or whether they've lost weight. So we have our different headings of our table. So our first heading is solute concentration in molar. Now we know from the past video tutorial that if it's got 0 solute concentration, then it's going to have a high water concentration. If it's got 0.4 solute concentration in molar, then it's going to have a low water concentration. So what we'd expect to happen is if we're putting a potato in a high water concentration, we'd expect it to gain mass and we'd expect it to become turgid. If we put it into a low water concentration, we'll expect it to lose mass and we'll expect it to become plasmolysed. So for the first part of this, you probably want to write down the table and all of the different questions. And pause the video. So the first part of this is to calculate the difference in potato mass in grams. So to do that, you need to take your mass of your potato at the end away, and then take your starting mass away from your mass of your potato at your end. So you would want to do 6.4 minus 5.6, and luckily enough, we are allowed to use calculators in biology. Right, then you would do 8.2 minus 7.8, and so forth. Right, to get your percentage change in your potato mass, you want to do your difference that you've just got, and you want to divide it by your mass at the start. And then you want to press the equals button in your calculator, and then you want to times it by 100 to get all of your percentage changes in mass. So what I'm going to do now is I'm going to move to the answer slide. So if we're working out our difference in mass, like we've seen in the other slide, we're going to do our end mass, so 6.4, take away our starting mass, so 5.6, and then we need to work out whether that is going up or if it's decreasing in mass. So 5.6 is a smaller number than 6.4, so obviously the potato has gained mass. So your difference in mass is going to be a plus value. So we write, and you need to remember to write the plus, so plus 0.8. Now, to calculate our percentage change in potato mass for this one, we're going to have to do our difference in mass, so 0.8, and we're going to have to divide it by our mass at the start, which was 5.6. And this is an important part because if you don't do this, then it will go wrong. You need to click your equals button on your calculator, so equals, and then you're going to times it by 100 to make it a percentage change. So if we've calculated that right, and remember our plus, we would have got plus 14.3%. So for the next one, we do exactly the same thing. So we have got our 8.2 this time, so 8.2, and we need to take it away from 7.8, and we want to work out what is the biggest number. So is it 7.8? No, it is 8.2. So our potato is gaining mass again. So our difference should be plus 0.4, remember that plus. And then we want to work out our percentage change in our potato mass. So again, we take our difference, so 0.4 we always put it over our mass or our original at the start. So that's going to be divided by 7.8. We need to remember to click the equals button in our calculator at this point, and then we need to times it by 100 to make it into a percentage. So remember that it's a plus, so it's going to be plus 5.1%. So looking at the next one, so this time we have got 5.1, our mass at the end, take away 5.4, so we need to have a look what number is bigger. So this time it's the other way around, so our mass was bigger at the start and it's got less at the end, so this means that it's going to be a minus number, so your potato has lost weight, so that's going to give you minus 0.3. So what you've got to do is in your calculator you put minus 0.3. We're going to divide it by our mass at the start, our original, which is 5.4. We're going to press our equals button. Remember that equals button, important. And then we're going to times it by 100 to make it into a percentage. And remember it's going to be a negative. So for this we should have got minus 5.6. Right, so for the next one we've got 3.9. And we're minusing it from 4.3. So we look for what one's the bigger number. And the bigger number is this one. So we are losing weight to get to 3.9. So it's going to be a negative number. So we've got negative or minus 0.4. So we're going to work out our percentage change. So again, that's your difference. So minus 0.4 divided by your original, which is your mass at the start, which is 4.3. Remember that lovely equals button, and then we're going to times it by 100 to make it into a percentage. So that will give us a minus 9.3%. Okay, so for the next one, we have 3.1 minus 4.3, and we have a look. So what number is bigger? It's going to be the one at the start. So that means that the potato has lost weight again. So it's going to be a minus or negative number. So it equals minus 1.2, sorry. So minus 1.2. Right, now we're going to look at our percentage change in our potato mass. So we need to put our difference, which is minus 1.2. We need to divide it by our original, which is our mass at the start. So that would be our 4.3. Click our trusty equals button on our calculator, then times it by 100, and we should have got minus 27.9%. And then the last one was done for you. For the next question, we have the calculation for calculating the percentage change in potato mass. So that's just asking you how you would do it, and you probably would want this noted in your jar. So you would have written that you put your difference in potato mass, you divide it by your mass at the start, and then you press equals, and then you times it by 100. So your difference divided by your starting mass times by 100. It's important that you know how to work out a percentage change in anything because it is a common question. So the next question is, what is the calculation for calculating the average percentage? So for calculating the average percentages, you'd have to add them all up, and then you'd have to divide them by the number there is. So you'd have to find the total. So add in this one to this one, 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 and this one to this one. Then you press equals button. Remember the equals button is always very important. and then you'd have to divide them by how many there is. So there is 1, 2, 3, 4, 5, and 6. So you divide it by 6. So it would be your total divided by your number of sets or groups that are there. The next question is, what is the average percentage mass of the potato in the 0.4 molar solution? So we need to find out where our 0.4 molar solutions are, which is here and here. And then we need to add the percentage change in mass, because that's what it's asking, the average percentage change in mass of the potato. So we want to add our minus 27.9, add our minus, so 27 plus 9, add minus, which would turn it into a minus 51.3, and we want to get that equals. Now, how many sets do we have? Well, we're looking at 1, we're looking at 2 things, so then we would get the total of that, and we divide it by 2. So our answer should be that divided by 2, which gives you 39.6%. Well done if you're managing to keep up, because a lot of things are tricky in these calculations. So you might need to go over the video a few times, or parts of the video a few times. So for the next question, why do we use percentages for comparison and not the maths at the end? This is a really good question because it comes up quite a lot. So whenever you get asked why you use a percentage and not the mass at the end, it's always because your values at the start are different. So you can't compare your values at the end because your values at the start are different. So you'd want to take the heading that's at the start and say that that is different. So for this answer for the question, it would be because the potato masses at the start were all different. So it wouldn't be a fair comparison unless you calculate the percentage change. Then question 4 is, in what solution was the cell most turgid, and how did the cell become turgid? So our answer for that should have been our 0 molar solution, and that's because it had the highest water concentration. So the 0 molar solution, the water moves from the high water concentration outside of the cell to the low water concentration inside of the potato cell by osmosis, causing the vacuole to swell and push the cytoplasm and the cell membrane against the cell wall, making it turgid. Then for question 5 it says, in what solution was the cell the most plasmolysed, and how did it become plasmolysed? So for this answer, it would be the 0.4 molar solution. And that's because the water moves from the high water concentration inside of the potato cell to the low water concentration outside the potato cell by osmosis, which causes the cell vacuole to shrink and pulls the cytoplasm and the cell membrane away from the cell wall, making the plant cell become plasmolysed. And for the last question, question number 6, it says, why was the cell blotted dry before and after it was weighed? Now this was to remove excess water, so to remove excess surface water which would affect the mass results, so to make the experiment valid. So summarising passive transport. So diffusion and osmosis are examples of passive transport because they do not require the input of energy. Molecules simply move from an area of high concentration to an area of low concentration, just like a ball or a cyclist going down a hill. It requires no energy input. So now we're going to learn about active transport. So your learning intention is to compare passive and active transport, to be able to define active transport, and to describe that the proteins in the membrane are responsible for the transport of materials. So for your success criteria, you'll be successful if you're able to label a diagram of active transport and provide the definition, if you're able to answer the questions on active transport, and if you're able to write down a comparison table of active and passive transport. So active transport is the opposite of passive transport. So sometimes molecules must be moved against a concentration gradient from a low region of concentration to a high region of concentration. So basically up a hill. So like if a cyclist had to go up a hill, they'd have to put in a lot of energy. They'd have to pedal that bike quite fast up that steep hill. So active transport is from a low concentration to a high concentration, and it requires energy. So we can remember that active is usually when we do exercise, and you'd be more active if you were riding uphill than if you were riding downhill. So you imagine a little person on a bike having to pedal really hard, put a lot of energy in. So active is up, so it's against the concentration gradient. So you're moving from a low concentration to a high concentration, and it requires energy. So you would want to write the heading active transport, and you'd want to put a note of active transport. So this is the movement of ions across the cell membrane against the concentration gradient from an area of low concentration to an area of high concentration. It is brought about by a carrier protein in the membrane, and it requires energy. So you need to learn this, you need to write it out a few times, you need to say it a few times. So you should be able to get this diagram from the consumables. So this is quite similar to the one that we looked at in the last tutorial about diffusion. So we have to look at where the numbers of the little molecules. So this would be the low concentration and this would be the high concentration. And now we're talking about active transport, and we know that it's the movement of ions from a low concentration to a high concentration. So the direction of the ions must be going this way in this picture. However, the one below, we've got our low concentration here, and we've got our high concentration at the other side. So this time it must be moving in the other direction. So you should be able to get that out of the consumables. So active transport into the cell, active transport out of the cell. So as long as it's moving from a low concentration to a high concentration and requires energy, it will be by active transport. And remember, it's these things called carrier proteins that are going to move the ions in and out. So this is an animation of these carrier proteins. So you've got your plasma membrane, which is made up of your phospholipid bilayers and proteins. So as you can see, your carrier protein takes your ions from your low concentration and puts them in the area of high concentration. So an example that you need to know of active transport is the sodium-potassium pump. So it's where a carrier protein exchanges sodium for potassium. So sodium is pumped out of the cell and potassium is pumped in against their concentration gradient, and it is important for the correct working of nerve cells and their correct transmissions. So here is a picture of the sodium-potassium pump. So you've got a high concentration of sodium ions on the outside of the cell and a low concentration of potassium ions. And inside the cell, you've got a low concentration of sodium ions and a high concentration of potassium ions. So what's going to happen is your sodium ions are going to get picked up by your carrier protein. From your low concentration, they're going to be moved to the high concentration. And then this then allows the potassium ions to come in, and they're going to be moved from their low to their high concentration. So again, your sodium ions going into your carrier protein, getting moved, and your potassium ions coming into your carrier protein. Inside. So you should be able to get this diagram, which tells you about your carrier protein. So step one is your free sodium ions enter the enzyme from within the cell, so they enter this protein channel. Then ATP phosphorylates the enzyme, causing it to pump the sodium out of the cell, which is against the concentration gradient. and moving from the low concentration to the high concentration out of the cell. Then this allows your potassium ions to come in. So 2 potassium ions come into the channel from outside of the cell where they've got a low concentration. And then the new unphosphorylated enzyme then pumps the 2 potassium ions into the cell where they've got a high concentration. Another real life example is is one of your CFTR proteins. So that is a channel, okay, and it's responsible for pumping out chlorine, which then is responsible for allowing water to be transported in, which then thins the watery mucus. So in cystic fibrosis, you've got a mutation in the gene which makes the CFTR protein. So the gene's found on chromosome pair 7. So basically this channel here doesn't work. And when this chlorine ion channel doesn't work, then the water can't get in. So you have a buildup of thick, sticky mucus and you've got more sodium ions getting out as well. So for this table here, you need to try and answer it. So pause the video and try to answer it on your own. So you want to give an example of a substance that moves through a cell by this process. You want to do the particles move from a high concentration to a low concentration or a low concentration to a high concentration. And is it down or against the concentration gradient and is the energy required? So you want to do that for diffusion and you want to do it for active transport. So if you pause the video now instead of me just telling you the answers. So for diffusion, an example of substances that move through a cell by this process, it could be oxygen into the blood vessels, it could be carbon dioxide out of the lungs. For active transport, we looked at the sodium-potassium pump in your nerve cells. And do the particles move from high to low or low to high? So for diffusion, it's always high to low because it's going down the hill. And for active transport, it's always low to high because it's going up the hill. Now for down or against a concentration gradient, diffusion begins with D, so it's always going to be down, and active transport is going to be against. And is energy required? If you're going downhill, you don't need energy, so no. And if you're going uphill, you need a lot of energy, so yes. So what you want to do now is you want to try the SQA PowerPoint, which is in your transport and in your PowerPoint. You want to try the cell biology exam practice questions, which comes in the teacher guide and cell biology exam practice questions, so page 6 to 8. You want to try page 9, 17 and 18 of the National 5 James Torrance textbook and also try your transport past papers. So use the questions and then see if you can understand whether you got them right or wrong and why. If you can't understand why, then email your teacher and try to figure it out. Then once you've completed all that, you can then try the cell transport homework. Once you've done the cell transport homework, email it to your teacher. So that is the key area 2. So transport finished. Woohoo! So on to key area 3.